The central dogma is the fancy term for going from DNA, RNA to protein. It's what turns information, which is in the form of your DNA, like what we always consider who we are, like you are your DNA. And it's the actual realization of that. It's what turns that into quote unquote reality, what you and I physically see on the outside. How is it that we turn these microscopic strings of random letters into your height, into your functional nails, into your hair? So this is what's going to discuss that. The first misconception I really want to address before we start is that when we say DNA turns into RNA, it really doesn't turn into it. It doesn't like convert and like your DNA is no longer DNA. It's now in the form of RNA. What I want to clarify and what we'll see in the further of this lecture is that DNA gets copied in instead of it being a copy of itself of being straight DNA, it's actually copied into another cheaper form, which is RNA. And then RNA gets read and is quote unquote copied again, but like more in the form of like a protein is the result of that copy instead of it being another strand of RNA. So let me kind of clear that up a bit. So we're going to start off in the nucleus, which I want to remind you is right here in the middle of the cell. And when you get in there, what we care about is that DNA is in there. Once again, these are your A, your T, your C, and your Gs. That's where you're going to see those codons come from. We'll discuss what a codon is, but these are the nucleotides within your DNA. It's amazing to think that these four letters are enough to dictate the entire entity of your being, how long your fingers are, how long your limbs are, what you're going to look like, your skin color, even your personalities. So this is where it all starts. Well, first thing you got to do is transcription. You want to turn these letters into reality. The first thing you need to do is you have to transcribe them. You need your DNA to actually be read by your cell. So that is going to begin in an area that's called the promoter region. This is a great name because this here is going to indicate where RNA polymerase is going to attach. RNA polymerase is what reads your DNA and makes that cheap copy I told you about, because your DNA is important. DNA is so important, we want to keep it safe inside of the nucleus. We want to keep your DNA safe in this little haven here. So what happens is RNA polymerase is going to attach to your DNA and make RNA, a cheap copy of your DNA, which if it gets destroyed, no big deal. So what's important to understand is this promoter region is very easy to find on DNA strands because that's where you're going to see a bunch of T and A nucleotides, aka the TATA box. And this promoter region is like indicating, it's like, hey, polymerase, come here, start reading the gene here. The gene starts here. And that can be anywhere on the DNA where you see this TATA begin. So transcription, this is when DNA is read, then copied in form of RNA. The DNA is still going to be there after this whole transcription event, but what's also going to be there is this RNA copy. All right, so how does that go down? Well, after the promoter region has been found by the RNA polymerase, which is being shown here as this big blue oval. As you can see, this is once again reiterating, once the polymerase goes through, it's not like, destroying DNA and reassembling it into RNA. It's simply unwinding your DNA. As you can see here, it's unwinding, right? And then this here is the RNA strand that's going to come jetting out of the side of it. Since you're already on YouTube, this after the end of this video, go and look up transcription. It's a fascinating thing to look at artist renditions of it and to see how it really can happen. It literally just shoots out of the polymerase as it unwinds and then rewinds your DNA. All right, so this here is still considered transcription. This is still here transcription. All right, this image here. All right, this is just a nice close-up view of it. RNA polymerase is very important to remember that RNA polymerase means that it makes RNA, not anything else. There's also DNA polymerase, but that occurs during DNA replication when you're copying your DNA to form more DNA. RNA is not really a process that is involved in DNA replication. It's only really prevalent here in transcription. All right. So what's the point of RNA? Well, like I said, RNA is a nice cheap copy of DNA. So it's okay for it to leave the nucleus. So the RNA, that red strand, is going to leave this nucleus here. Right there it is, leaving the nucleus, and it's going to travel around until it hits a ribosome, which is not pictured somewhere here. But eventually it will hit a ribosome somewhere in the cell. And it's okay if it gets lost on the way there because your DNA can just be recopied or reread. And once it hits that ribosome, a ribosome, remember, let me write this much neater, slower. A ribosome is the organelle that is going to translate or make proteins. We always talked about that in the beginning of the chapters, right? Oh, it makes proteins when you're making your cell cities or whatever. But now we're going to see how it really does make a protein, because these proteins can range anywhere from hair to nails. How do we make sure that we don't have nails growing out of our nose hairs, right? Or something like that. Well, it's because ribosomes are read that special code, that RNA strand that's specifically coding, oh, we're making nose hairs today. And that's what the ribosome is going to do. So ribosomes make proteins by reading codons. Let me rewrite this. I have an issue with my handwriting. By reading codons on RNA. All right. So, and to be specific, this is mRNA. M stands for messenger. So once the ribosome reads it, this is an image of what it looks like. Here, the ribosome, what you'll see is it reads it in codons, which is 3 RNA strands. Let me actually highlight it so it's more clear. These 3 strands here, I'm going to circle it as well, just so it's easier for anyone who wants to see it. That's a codon. And based on that codon, it's going to pick a particular amino acid. You see, this here is an amino acid specifically set for this codon here. And then this next codon here is going to trigger for this amino acid to be added. All right. And it's added in the ribosome in what's considered the P site. And there's really good images online as well. There's 3 sites within the ribosome. There's E, P, and then A. P is where the strand is attached. A is where it comes in. You see how these these things here, these are called tRNAs. Here it is right there. There's a name for it right there. tRNAs, these big squiggly things. Not to be confused with mRNA, but these transfer the amino acid to this growing chain. So these tRNAs go to the attachment site, the A site. They put the peptide or the amino acid onto the peptide chain in the P site, and the E site is the exit site. So exit site, attachment site, or attachment site. And then P, the P site is where the peptide grows. So these are all great things to have in your notes in regarding to what's going on inside of the ribosome. All right, once again, the ribosome, it reads this RNA strand, this mRNA. You're going to really want to be sure you know the difference between these two. The mRNA that just came from the nucleus is being read in the ribosome, so tRNAs know exactly which amino acid that goes on there. And this whole process here is called translation. You're translating the script that was made in transcription. That's translation. Making protein based on mRNA message. Okay. Ooh, misspelled message. I'm just going to abbreviate that so I don't take any more time. Okay, now we get to something that's going to confuse a lot of students, and that's how you read these RNA codon charts. Every one of these letters represents the RNA codon that is that you see here. You see all these circles that I drew here, right? That's a codon. That's a codon. That's a codon. These here are the representing a codon. And each codon is going to specify a specific amino acid. All these blue sets of letters here, including this green one, these are all amino acids. So these are codons, but these are amino acids. And each codon specifies a specific amino acid to be read. The type of question you can get is, let's say that you have a DNA template strand reads, let's say that it reads A, T, G. Well, then that means the RNA strand, let me actually section this off so it's easier. The RNA is going to read as U, because there's no T for RNA, A, C. Well, what does our UAC code for? So the first letter is U, second letter is A, and the third letter is C. So UAC is going to code for TYR. I want to say that's tyrosine. All right, so protein. I could be wrong, but it's going to be TYR as the amino acid. So that's the type of question that your teacher can give you. That, oh, here's a DNA template. Here's a codon from the DNA. What's the RNA going to look like? Therefore, what is the protein going to be?